right, good afternoon. Welcome to everybody here in the room and to those of you who are joining us via the webcast. My name is Jeffrey Kahn. I'm a professor of bioethics and public policy at Johns Hopkins University, and I had the privilege of chairing the Committee on the Ethical and Social Policy Considerations of Novel Techniques for Prevention of Maternal Transmission of Mitochondrial DNA Diseases. A mouthful still. We're really pleased to be here today at a public discussion event. So the sort of order of things here, just to give folks a sense of what we're going to do, is first, once I finish my welcome and a little overview remarks, I'm going to turn it over to Celia Witten from the FDA, after which the committee will do a short presentation about our recommendations, and then we will open it for questions here in the room and via the webcast. So we will devote a fair amount of time to discussion, which is, of course, the purpose of our being here. So I think without any further ado, I will welcome and thank Celia Witten, who is the Director of the Office of Cellular, Tissue, and Gene Therapies within the Center for Biologics Evaluation and Research of the FDA.
Thank you for that introduction and for coming here today. What I'm going to do is I'm going to present the same presentation, just with a little bit more, that I provided to this committee at the outset of the study project that in the meeting where we set the charge for the committee. But before I do that, I just would like to publicly thank both the committee and the IOM staff for what really was a wonderful job to resulting in a report in an area which must have been very complex and challenging for both the IOM staff and also for the committee. So thank you. So just as I said months ago or a year ago when this project started, so to set the stage for this, the problem is the following. There's a woman with a mitochondrial disorder based on mitochondrial disease that transmits the disease to her first child. She wishes to have a second genetically related child, but without transmitting the disease. What are her options? And I'm not going to go over this in detail. As probably many of you know, the mitochondria are energy-producing organelles, subcellular organelles, and they have a number of other functions as well. Mitochondria contain their own genome, and these are inherited maternally. There's a high mutation rate and a number of known diseases from mitochondrial mutations. So what brought up the whole, the interest in this from our end was the growing scientific literature looking at two approaches to help solve the clinical problem that I presented, which is avoiding maternal transmission of diseased mitochondria. One is so-called maternal spindle transfer and then pronuclear transfer. I'm not going to go into these in detail, except that the maternal spindle transfer involves a donor oocyte with the spindle removed and replaced with the spindle from the mother, and then subsequent fertilization to reconstitute an embryo. And pronuclear transfer is similar, except that the pronuclei of the zygote are removed and replaced with the pronuclei from the parents and the embryo reconstituted in that manner. So knowing that the questions were coming regarding what would be needed in order to support an investigational study in this area, our office held an advisory committee meeting in February 2014, and I have the topic listed: mitochondrial manipulation technology and assisted reproduction for prevention of transmission of mitochondrial disease or treatment of infertility. That meeting, the stated purpose of that meeting was to discuss potential future clinical trials of mitochondrial manipulation technologies. It was not intended to focus on a specific technology. No specific clinical trial was discussed. and it wasn't intended to support a specific regulatory action. So in other words, it wasn't because we had a specific file that we were taking action on. It was to prepare ourselves with the background of a scientific discussion regarding what we should consider in an application of this nature. Specific topics discussed at the advisory committee in 2014 included ability of animal models or other methods to address safety issues, risks to mothers and children, clinical trial design elements, and manufacturing and processing controls for safety. Not discussed, so at the meeting, scientific issues were discussed, and that was what the meeting focused on. Not discussed at the meeting were ethical and social policy issues raised by heritable genetic modification. We received a number of comments from the public at that meeting, and not only because of those, but just because we also realized that these were important concerns for the public, determined that we would ask the Institute of Medicine to do the consensus study that is the subject of today's discussion. The next couple of slides are from this verbatim from the charge to the committee. They're excerpted from the charge to the committee. So ethical and social policy issues, these were examples of issues that we asked the committee to address, but we didn't restrict them only from addressing these specific issues. So one is whether manipulation of mitochondrial content should be considered germline modification from a social and ethical perspective, the implications of manipulating this content, both in children born to women as a result of participating in these studies and in descendants of female children, ethical issues in providing consent for risks on behalf of a child who doesn't exist, and ethical and social issues for a child born with genetic material from 3 individuals. One of the questions we asked the Institute of Medicine to consider was the foundational question of whether safeguards such as specific measures and public oversight could adequately address these social and ethical concerns, or whether the concerns precluded clinical trials. And then we asked for specifics on some specific issues that we wanted them to address if a trial were to go forward, such as informed consent, enrollment, and tracking, how we should take into account alternative approaches or factor this into our assessment of a trial, and whether we should, or what we should consider in terms of what the implications would be for trials that might go beyond these specifics under consideration, which is prevention of transmission of mitochondrial disease. So one of the questions that came up that we discussed at the Institute of Medicine kickoff meeting, so to speak, is how we were going to use the information. And one of the things that I explained at the time was that when we receive an application for a clinical study, we have 30 days either to let the IND be in effect or to put it on hold. That review is confidential. And certainly for reviews of applications in this area, we'd want scientific and ethical advice so that we were preparing ourselves for review of these types of applications, both by having our advisory committee to discuss the scientific issues and the Institute of Medicine consensus report to consider the social and ethical issues also in our application review. So that's where we left off during the meeting in which I introduced the charge to the committee. Subsequently, though, I want to mention that in December 2015, Congress passed and the President signed into law the Consolidated Appropriations Act of 2016, known as Public Law Number 114-113, which for fiscal year 2016 prevents the FDA from using funds to review applications in which a human embryo is intentionally created or modified to include a heritable genetic modification. As a result, the agency will not accept or review INDs related to genetic modification for the prevention of transmission of mitochondrial disease in the fiscal year 2016. I have the language from the law verbatim on this slide so that people can read it for themselves. One comment I want to make, and it just relates to the language in the law, it's the question of what it means to not receive an application. What it means is the IND will not be accepted for review by the agency, and because FDA will not accept the FDA application, human subject research utilizing genetic modification of embryos for the prevention of transmission of mitochondrial disease cannot be conducted in compliance with the Federal Food, Drug, and Cosmetic Act or the Public Health Service Act and FDA's implementing regulations. So that concludes my talk, and now I'd like to turn it back over to Dr. Khan for the committee's presentation.